Hello, and a very warm welcome to today's ISNTD Connect. My name is Marianne Compret, and I'm speaking from the International Society for Neglected Tropical Diseases. And it's going to be my great pleasure over the next 90 minutes or so to join you for this session. Now, today's meeting is the next session in our series on all things schistosomiasis in partnership with the Global Schistosomiasis Alliance, or the GSA. And we're going to be taking a closer look at the incredible journey and achievements of the Philippines in the control of schistosomiasis, focusing on Schistosoma japonicum. This promises to be an incredibly rich session. We are going to hear about the progress made since the advent of MDA in the 1980s in reducing disease prevalence dramatically, but also learn about the incredible work tackling the range of ongoing challenges which remain, whether in terms of the intermediate snail host, the threat of the carabao or water buffalo as a zoonotic reservoir, vaccine development, and more recently, even the COVID-19 pandemic. So a really rich session ahead of us with lots of learnings transposable to other regions, groups, and challenges. And so it's both our pleasure and, in fact, our honor to welcome today's speakers to take us through this vast range of issues and share their invaluable lessons. So I'm sure you will join me in welcoming very, very warmly Dr. Lydia Leonardo, first and foremost. Good afternoon, Lydia.
Hi, it's a pleasure to be here. Thank you for inviting us.
We're so happy to have you here today. Dr. Lydia, you are Professor Emeritus at the University of the Philippines. You're also Director of the Office of Research Coordination at the University of the East. And I think it's an understatement to say you've got tremendous experience of running schisto control programs. To be honest, with experience since the early 1990s, and you've also got knowledge on the programs at the Philippines Department of Health. So thank you very much for being with us today. We're also joined by Dr. Thank you, Lydia. We're joined by Dr. Mario Antonio Giz II. Hi, Mario.
Hello, good morning.
Hello.
Good afternoon, good evening, everyone.
Thank you, thank you. Mario, you're the head of the Immunology Department at the Research Institute for Tropical Medicine, and you've been working on, amongst many other things, vaccine development for Schistosoma japonicum, and very interestingly, the use of the local water buffalo that I mentioned earlier on as an animal model for this vaccine. So we'll be hearing more about that in a few moments. So thank you for joining us today. And also welcoming you very warmly, Dr. Ian Kendrich Fontanilla. Hello, Ian.
Hello, hello.
Hi. Dr. Ian, you're professor at the Institute of Biology at the University of the Philippines, and you're going to be talking to us a little bit later on about the snail intermediate host of Schistosoma japonicum, Oncomelania hupensis quadrata. So we're looking forward to that, and particularly your findings in terms of surveillance and monitoring of these snails and the growing evidence of their spread to other areas. So a really packed and rich session, absolutely looking forward to it. And without any further delay, let's move on to the presentations. For the interest of a smooth running of the webinar, we have got some pre-recorded presentations. So I'm going to now play Dr. Lydia, your presentation, and you'll be talking to us about sorry, I'm just getting that video queued up. Dr. Lydia, you'll be talking to us about schistosomiasis in the Philippines. The struggle never stops. I like that title.
Yeah, thank you.
And over the next few minutes. Over to the video.
Greetings to all of you who are watching this webinar today. I'm very grateful to the Global Schistosomiasis Alliance and the International Society for Neglected Tropical Diseases for taking interest in Schistosomiasis japonica, particularly in the Philippines, and inviting our group composed of Dr. Mario His from the Research Institute for Tropical Medicine in the Philippines and Dr. Ian Kendrich Fontanilla from the Institute of Biology of the University of the Philippines, and myself to present to you various aspects of this public health problem in the Philippines. I will take care of the current situation of the disease in the Philippines. The Schistosomiasis Control Program Manager is supposed to take charge of this aspect, but he's buried deep in work and has asked me to present the program in his behalf. Dr. Hies will talk about the reservoir hosts of Schistosoma japonicum in the Philippines, and Dr. Fontanilla on research updates on the snail intermediate host. Schistosomiasis in the Philippines was first reported by Dr. Paul Woolley in an inmate at a national penitentiary in 1906. This report was subsequently followed by many more cases in the same penitentiary where the inmates are coming from various parts of the country. This is the life cycle of Schistosoma japonicum, showing how integrated it is with the daily activities of the endemic communities, such as the daily chores of laundering clothes, washing of dishes, and bathing. The workplace of farmers, fishermen, and irrigation workers are also active sites of transmission of the disease. These are severe cases of schistosomiasis that should not have reached this stage had treatment been given early. Sad to say, even with an effective drug and with effective interventions, there are still people who succumb to the disease in endemic areas. This case is a farmer diagnosed positive for the disease and whose ultrasound showed a cirrhotic liver. He has been a farmer all his life and confessed to never having participated in MDA because he thought there is no point since he will be repeatedly reinfected because of his exposure in his farm. Schistosomiasis has a strictly focal distribution in the Philippines, confined in areas where there are snail intermediate hosts belonging to the species Oncomelania hupensis quadrasi. These areas are characterized by continuous rainfall all throughout the year with an indistinct dry season. The disease is perpetuated by poor environmental sanitation, inadequate supply of water, contact with contaminated water, and presence of infected mammalian reservoir hosts, such as water buffaloes, dogs, and rats, among others. The 28 endemic provinces have been stratified as high prevalence, moderate prevalence, and low to nearing elimination level. Overall, there are 12 of the 17 regions, 28 of the 81 provinces, 196 of the 1,489 municipalities, 15 of the 145 cities, and 1,609 of the 42,306 barangays that are endemic to schistosomiasis. It is estimated that 3.4 million people living in endemic areas are directly exposed to disease transmission sites. The stratification of endemic areas is now determined at the barangay level or village level. A barangay is equivalent to a village with population of 5,000 or more. In the past years, the national program, together with partners, have been doing focal surveys to determine the prevalence of each barangay. Because of the focal nature of the disease, the purpose of the focal surveys is to stratify the barangays based on the level of prevalence, which in turn will be the basis of the package of interventions customized for each barangay. As the disease prevalence goes down and the program succeeds, adjustments in interventions are made towards an approach that is more focused on residual areas of transmission and combination of interventions. For example, MDA combined with other public health interventions. The next slides show the actual situation in endemic areas and explain the difficulties in the control of schistosomiasis in the Philippines. The inadequate water supply forces people to do their laundry and bathing in contaminated water, exposing them to infection with Cercariae. Because of lack of toilets, people defecate anywhere, aggravating environmental contamination with schistosome eggs. Children mostly get infected by bathing and adults by performing domestic chores in contaminated water. Traditional method of farming uses carabaos, which are very good mammalian reservoir hosts. The use of rice varieties that require too much water also contribute to perpetuation of snail habitats in rice fields. These types of water supply create poodles of water that can support snail populations. Wastage is high because water flow is not regulated. The importance of water capture technology and bringing water to households has been reiterated in many consultative meetings in the past. In many poor endemic areas, the kind of toilet or latrine leaves much to be desired. Many are not water sealed, and if they are, the lack of water supply forces the community to eventually abandon the use of communal or public toilets. Perpetual flooding in many endemic areas expose the villagers to schistosomiasis. Heavy rains can cause floods or overflow from rivers during high tides, flood residences found along banks of rivers or streams. The snail habitats are just right there in their homes. The next slides show the various strategies and activities in the current control and prevention programs for schistosomiasis. In the Philippines, the schistosomiasis control and elimination program's overall goal is to eliminate schistosomiasis as a public health problem in all 28 endemic provinces using various strategies such as preventive chemotherapy and infection control, transmission control, public-private partnership, advocacy and social mobilization, and monitoring and evaluation. The goal in high to moderate areas is to eliminate morbidity through MDA of the exposed population. Once the prevalence has been reduced significantly to very low levels, this status will be sustained through strengthened active surveillance of humans and snail vectors, infection control, transmission control such as by sanitation and hygiene by way of health infrastructure, safe water supply, water-sealed toilets, and ordinances to control animal hosts. Mass treatment, especially among school children, and quality control of laboratory and laboratory staff. To date, 2 provinces have not shown cases in fecal surveys to determine prevalence. Partnership is being promoted to solve the problem of schistosomiasis. Public-private partnerships is expected to address various aspects of the disease. Some initiatives in the past have been undertaken to show that significant progress can be achieved in the control and prevention of schistosomiasis. Gonzaga in Cagayan Valley was the site of a multi-country project of the International Development Research Center of Canada, which aimed to develop the eco-health approach in the control of schistosomiasis. This approach intensified the multidisciplinary, multisectoral, and multi-agency involvement in the control of schistosomiasis in the Philippines. CONCAN was a part of this multi-country project, and this approach was a huge success in the Lawa model of the control and prevention of Opisthorchiasis viverini in Thailand. In transitioning from control to elimination, these challenges have to be overcome. A surveillance system based on highly sensitive diagnostic technique is essential. Capacity building has to be intensified for surveillance works. On top of these challenges are changes that cannot be stopped and seem to be railroading control efforts, such as construction of dams, floods, deforestation, and global warming. When the pandemic struck early last year, WHO issued pronouncements on postponement of NTD surveys, active case finding activities, and mass drug administrations, all of which involve people gathering together in one place. or very close physical interaction among people. There were activities that were encouraged to continue, such as prompt diagnosis, treatment, and essential vector control to prevent any further exacerbation in the NTD status, in particular areas like those with high prevalence and high intensity of infections. From the status report of Dr. Winston Palasi on schistosomiasis in the Philippines during the Schistosomiasis Control and Elimination Program Forum held in September 2020, he enumerated the following effect of the pandemic on NTD programs in the Philippines. Following WHO advisory, there was cancellation of NTD plans, programs, and projects implementation, and delay in the implementation of ongoing plans and activities of the NTD programs. The so-called COVIDization of programs was reflected in the mobilization of the technical staff and repurposing of office line function for COVID-19 response and realignment of programmed NTD funds and resources to support response to COVID-19 operations. In the midst of the pandemic, measures that have been in place for NTDs were further intensified to fight COVID-19. These include WASH, the use of the NTD surveillance mechanism, and health promotion and education platforms. While the COVID-19 pandemic, compounded by calamities and disasters in the country, no doubt stalled the progress in the control and elimination of schistosomiasis and possibly even offset the gains of the program. Some program implementers went out of their way to even innovate to improve service coverages. Good practices that led to wider MDA coverage included house-to-house approach, distribution of groceries, especially at a time when many households could have reduced incomes, frequent orientation to respond to questions about MDA, strong support from the local government units, strong partnerships with NGOs, and more extensive information dissemination. The Philippine Department of Health will complete the design and development of a national policy and guidelines on the control of animal schistosomiasis in close collaboration with the Department of Agriculture, Bureau of Animal Industry, using the One Health approach. Stratification of endemic areas will be continued and completed, and designing the conduct of MDA in the midst of the pandemic and in preparation for future emergencies. Prior to the pandemic, the schistosomiasis program has been confronting challenges. Climate change is one which continues to impact our lives, particularly our health. Nonetheless, there have been good practices that have pushed the program forward in its elimination efforts. Given all of this, in the end, how the Philippines will get the upper hand over this disease will depend on how strong the forces of our challenges will pull us back vis-a-vis the advantages that we have at present that can push us forward. We have better approaches to schistosomiasis control, hopefully covering all aspects of schistosomiasis, such as biomedical, social, cultural, economic, behavioral, and ecological, that will help us develop better intervention measures. We know we need better technologies, and we are developing them to track down the remaining holdouts of schistosomiasis in order to reach elimination. We develop models that can guide us on proper combinations of interventions in various epidemiological settings. As a result of the pandemic, we have established more development partners, investing in WASH and even in primary health care. Research is nonstop, whether basic, applied, or operational. Lastly, we have people who have made it their mission to work until the last trace of schistosomiasis can no longer be recognized as a public health problem or ultimately eliminated. Therefore, so much rests on our decisions at present. As we transition from emergency to the new normal, we make sure we learn from the hard lessons thrown at us in this pandemic. We make sure that this new normal is a better one, that our new normal schistosomiasis program is resilient, and can endure the challenges it encounters, and at the same time integrate, modify, and adjust according to demands. Let us also not forget that we have sustainable development goals to achieve that should not be forgotten or set aside as we all journey to recovery. So thank you very much for your attention.
Well, thank you very, very much, Dr. Lydia. That was an amazing presentation, actually, just reminding us, on the one hand, of the severity of a schistosomiasis infection, what it can mean to individuals, and the complex interplay of factors that can lead to reinfection, infection, but also, on the other hand, some really encouraging points about very forward-looking points about the future. So it'd be really interesting to discuss and elaborate on that in our discussion later. Thank you so much for this very comprehensive overview. And one thing that perhaps might be interesting, I know our audience is very varied, lots of professionals and researchers in schistosomiasis, but also perhaps from other disciplines. And maybe as and when you can, if you're getting involved in the chat, which is the small icon just around our video feeds here, if the participants as well could maybe throw in a few thoughts or a line about how has COVID affected the programs that they work on. Anything big or small would be really interesting to know. And I think today's all about learning from this very interesting and invaluable example of the work of the Philippines, not just for schisto control, but NTD programs in general as well. So please, by all means, do go ahead in the chat. Let us know what have been your experiences with COVID. And I'm sure Dr. Lydia, our speakers, and ourselves will be really interested to hear about that. We'll come back to that in the discussion. But for now, Dr. Ian, so your presentation was pre-recorded as well. So we're going to try our luck with a second video. And you're going to be taking us through some of your work on the snail intermediate host and your surveillance efforts there.
Hello, good morning, good afternoon, good evening, wherever you may be. I am Ian Fontanilla from the Institute of Biology, University of the Philippines, Diliman, and I'm here to present our university's contribution on the current research on Oncomelania hupensis quadrasi. So I will also show the relevance and implications towards target of schistosomiasis elimination in the Philippines. So the work we will present here are those covered by my projects together with my colleagues at the Institute of Biology, as well as those from the other constituent units of the university. So at the outset, I would like to take this opportunity to thank the Global Schistosomiasis Alliance and the International Society for Neglected Tropical Diseases for giving us a platform to present our work. Okay, so let's start. So the outline of my talk will include the following. I'll give a brief introduction of the snail, as well as show, and then I'll move forward immediately to our work. So we have some malacological surveys on the snail, particularly on the infection rate, prediction of snail habitats, and the effect of meteorological events to the snail dispersion. And then I will also focus on eco-epidemiological surveys involving the snail, particularly with the use of eDNA as a proxy for biosurveillance. And then the last part will focus on the genetic aspect of the snail, with emphasis on phylogenetic and barcoding studies. So I'm sure everybody knows that one of the snail intermediate hosts of Schistosoma japonicum is Oncomelania hupensis, particularly the Oncomelania hupensis quadrasi, which is found here in the Philippines. It belongs to family Pomatiopsidae, and it is an amphibious and dioecious snail. It's a very small snail, around 3 to 5 millimeters only, and it's quite difficult to detect them in the wild, especially if you're not a trained malacologist. In the Philippines, there are around 3,012 identified snail-infested bodies of water, and around 80% of these infested bodies of water are found in Mindanao. So we have yet to cover the different areas of the Visayas and Luzon group of islands to see if some of these bodies of water could potentially be could potentially harbor the snails. and more importantly, the parasite. So this is a typical habitat of the snail. Usually they live in swampy, murky water with usually turbid, sluggish moving waters, etc. And I will give more emphasis later on when we try to characterize the habitat of the snail as an as a means to identify potential risk areas. So this shows you a picture of how small the snails can be. Now, there are other Oncomelania hupensis subspecies, and usually they're classified based on their morphology, biology, and even their antigenic characteristics. In China, there are four major subspecies. So OH hupensis hupensis, OH robertsoni, OH tangian, and OH guangxiensis. In Japan, there's one. In Indonesia, there's one. And in Taiwan, there are two possible subspecies. As I've mentioned, in the Philippines, we have O. hopensis quadrasi. Now, in the Philippines, we have a goal towards schistosomiasis control and elimination. So the target is the interruption of transmission of schistosomiasis in all endemic barangays or villages by 2025. So that is a little less than 4 years from now. So let's see how we progress. By definition of transmission interruption, we mean the reduction of the incidence of indigenous human and animal infection to zero, okay, and the reduction of snail infection also to zero. So a very important approach towards the achievement of this control and elimination is a surveillance of the snail intermediate host. And in particular, we want to interrupt the life cycle of the parasite at the level of the snail. Okay. However, surveillance in many endemic countries, the Philippines included, is dependent on microscopy, which has a very low sensitivity, particularly for low density infections, because the particular stages of the parasite, particularly the cercaria, the sporus, and the sporocyst, they are very, very difficult to identify or to obtain and identify from the field, particularly because these are environmental samples. You will find them in the environment, either in the water or in the snail. Okay. And then another approach for biosurveillance is a sentinel mouse technique, wherein you expose some mice into potential risk areas like bodies of water, in the hope that the cercaria will infect the mice, and then after 35 days, we will see the emergence of the adult worms. However, this detection method has a certain delay of around 35 days. So by which time it could be, the spread of the time that could have been allocated to control the dispersal would have been lost. Okay, so at least in the Philippines, there is a need to determine the current state of snail infection, identify the environmental factors that affect the dispersal of the snail intermediate hosts, and to optimize other methods for snail detection. In particular, we're going to focus on the environmental DNA or eDNA, and then to assess the genetic diversity of snail populations for possible role in susceptibility to infection to Schistosoma japonicum. Okay, so for the first set of studies, I will focus on malacological surveys that we have conducted or our colleagues have conducted. And the first one is a malacological survey of endemic villages in the Philippines. So this paper was published with which was a part of a master's thesis by Mr. Rafi Fornillos, together with our collaborators from Japan and Dr. Dr. Lydia Leonardo. Excuse me. So we conducted a typical, when I say we, Rafi conducted the malacological survey to sample to the different sites, and then sample confirmation, identification, and then snail crushing to determine infection. And what we have found is that from 38 snail sites from these provinces, we recovered 6,279 snails. And the infection rate varies across different sites. And even within the same site, within the same village or the same province, we have a range of infection. So we get an infection rate of 0%, to as high as 14.71%. Now, this is significantly high. So as you can see, there is a patchy distribution of, there's a patchy infection, rate of infection. Even within the same site, it could vary across different snail populations. And the differences could be due to the degree of fecal contamination and their connectivity to water that can serve as contamination source. Okay, so uh, so if you will recall, schistosoma life cycle it has to be exuded through the feces from the definitive or final host, and then the infection will the snail will eventually be infected in its follow if it follows through the life cycle. Okay. So one interesting thing that we noted is that the Bohol seems to be a candidate for elimination because previous studies have also shown zero infection, at least for clinical samples. So we will have to focus on Bohol for the next succeeding surveys, both for malacological and definitive or final host surveys to determine if Bohol is indeed a candidate for elimination for schistosomiasis. Okay. Now, the next study conducted by our PhD student, Dr. Daria Manalo, aimed to predict the snail habitats of Oncomelania hupensis quadrasi and identify the risk areas for Schistosomiasis japonica in one endemic region, Gonzaga in Cagayan, which is one of the latest sites in the Philippines which have been designated as endemic for the parasite. So in 2002, this was first reported by Belisario et al. And then in 2004, one barangay was determined to be endemic. And then 4 years later, this has increased to 3 endemic barangays. So Tapel, Magrafil, and Santa Maria. So there is a concern for Gonzaga because there seems to be a spread or dispersal of the snail and the parasite that it harbors. So there is an urgent need to determine how they disperse, identify the potential risk areas, and mitigate their dispersal. Okay. So we need to develop a methodology that is feasible and useful to be implemented in other endemic areas that will help identify risk areas or potential snail sites that may harbor the parasite. So they conducted extensive malacological survey in several barangays in Gonzaga, and then aside from that, they measured the GPS and then determined the ecological parameters, environmental parameters for each site in order to correlate the ecological parameters with the presence of the snail. Okay, so what we have found out is that the potential snail sites can be characterized by the following: topography, usually at lower elevation, with spring or swamp habitats or swampy habitats, excuse me, and then the soil is the silt loam type. Okay, so majority of the snails are found in the silt loam soil type, and usually, if you have a previously known snail site, the closer the new site is to the previously known snail site, the higher the chance that it will also harbor the snail, which is logical. And then furthermore, water tends to be sluggish, and they have high level of soil organic matter as well as low level of water ammonia. However, there are these parameters are not statistically significant predictors of potential snail sites. So they're not significantly different compared to the other sites which can also harbor the snails, but they can be characterized by these features. So there must be another or additional parameter by which we could identify the snail in potential risk areas. And there has been a recommendation that maybe aside from identifying the physicochemical parameters of sites to predict risk areas, maybe we should also use environmental DNA. Okay. Now, another study conducted by the dissertation student of Dr. Lydia Leonardo, Dr. Gracia Varona of UP Visayas, tried to determine the effect of of meteorological events, in this case, a super typhoon, on the distribution and dispersal of the Oncomelania hupensis quadratis snails. Just a brief background, Typhoon Haiyan, or locally known as Yolanda, hit the Philippines, particularly the island of Palolete, in November 8, 2013. Now, this one is a super typhoon with a wind speed of up to 350 plus kilometers per hour. So it's a very, very powerful tropical cyclone, tropical typhoon, and it has caused significant damage. Now, when Dr. Gracia tried to evaluate the presence and distribution of the snails as well as the infection rate, she found out that among the 33 barangays covered from November 2015 to October 2017, so this is 2 to 4 years after the typhoon hit, and when she compared her results on historical data, she noted the absence of snails in coastal barangays, which were previously listed as barangays positive for snails. And then she also discovered snails in some villages or barangays, such as Teranza and San Isidro, which were not included in past reports as positive for snails. So some of these snails were dispersed to these areas. And she also detected infected snails in sites which were previously reported to harbor only uninfected snails. Now, the results of these, although we cannot say positively for sure that super typhoon has caused the dispersal, but it is possible that these and other typhoons could affect the distribution of snails into new areas. And with them, they could potentially carry the trematode parasite. Now let us focus on eDNA studies. Now, eDNA, or environmental DNA, are DNA that are extruded by organisms into the environment, which they could come from sloughed off cells, or dead cells, decomposing cells, etc. And they could be used as proxy for the actual specimen itself, or especially if they are very difficult to find. And this will also limit the risk to the collectors or to those that are involved in biosurveillance, especially if the organisms they are targeting are potentially of high risk for example, for disease like schistosomiasis. Okay, so our first study here, this was published by Rafi as part of his master's thesis, wherein he tried to develop or optimize eDNA detection of schistosoma as well as oncomelania from water samples. So we based our method on the cytochrome oxidase subunit 1 gene, or COX-1 gene, which is a mitochondrial gene. And it comes in high copy numbers precisely because it is found in the mitochondrial genome. And then we have a lot of mitochondrial genome copies within each cell. Okay, so this is the methodology. And then these are the sites that we surveyed: Northern Samar, Leyte, and Compostela Valley. And then this is just to show proof of concept that indeed we were able to target the DNA in the lab for both eDNA for both Oncomelania hupensis quadrasi and the Schistosoma japonicum. So here, we were able to detect the DNA of Oncomelania hupensis quadrasi, and in the presence of other snails like Limnea, Pomacea, and Melanoides, the probes or the primers did not anneal or amplify the DNA from these snails. And then we all likewise, we were able to do the same for Schistosoma japonicum. We were able to target the DNA of Schistosoma japonicum, but not other trematodes like Paschola. Okay, so this shows you some sample qPCR amplification plots. This one for Schistosoma japonicum. So this one's the control, and these are the laboratory water samples and field samples that contain the Schistosoma japonicum, and then this one for Oncomelania hupensis quadrasi. Now, interestingly, we obtained results from the field. So we obtained water samples from the field, and these are the different sites, okay, snail sites. Now, for sites where no snails were observed, when we went there, when our collectors went there and tried to look for the snails, and they did not find any snails within a prescribed period of time. Interestingly, when we collected water samples from there, we were able to detect that the eDNA of Oncomelania hupensis quadrasi. Therefore, we can be certain that the eDNA of if the eDNA is there, then there's a possibility that the snail itself is there or from a nearby area, except that we did not detect them manually or physically by eye. And this is very important, especially for biosurveillance, because malacological surveys can be prone to mistakes wherein the collectors were not able to observe the snails. And it's also very risky because they themselves could be infected by the parasite. And then interestingly, we detected also the presence of the eDNA of Schistosoma japonicum, even in supposedly these areas where there is no snail present. Now, when there are snails present, ayan, interestingly, when we detected the snail physically by eye, in almost all cases, we also detected the eDNA of Oncomelania hupensis quadrasis, except for one site. Now, again, this could be possibly due to the patchy distribution of the eDNA. Remember, eDNA is not homogeneously distributed in a water sample in the same way that the snail itself is not homogeneously distributed in a particular area. And then, interestingly, we also detected the presence of Schistosoma japonicum, even if in these sites wherein infection rate is non-detected or not detected, meaning despite obtaining the snails and despite the fact that we did not find the Schistosoma japonicum microscopically, based on the lab techniques we had, we did not detect the Schistosoma japonicum cercariae, but we were still able to detect the eDNA, so they were still present. So we were able to successfully demonstrate the utility of the cytochrome oxidase subunit 1 gene for both the Schistosoma japonicum and O. hupensis quadrasi, and the primers used have been proven to be specific for both species. So in this regard, malacological surveys could be complemented with eDNA detection for both snail and the parasite. In fact, this could be an easier method of detection and far less risky compared to the traditional malacological survey. Now, moving forward, we tried this method using soil samples this time. And going back to Gonzaga in Cagayan, we obtained soil samples and from different areas in Gonzaga, and taking note also of the physicochemical parameters. So we combined eDNA detection as well as an environmental approach. Okay. So we obtained 27 sampling points and 3 replicates per sampling point for a total of 81 soil samples. And then from there, we obtained extracted the eDNA and tried to detect the presence of the snail. Excuse me, of the snail. Now, this is a pioneering study here in the Philippines that used eDNA to detect the snail from the soil, and we were successful in doing that. And we found out that we could recover eDNA from as much as 6.5 meters away from water bodies. So these snails, apparently, if we're going to use their eDNA as proxy, they can be found as far as 6.5 meters away from bodies of water. And the recovery of eDNA increases as the pH, phosphorus, zinc, copper, and potassium content increase in the soil. And the distribution mapping confirmed that the snail can extend their habitat range at high amounts of elemental soil nutrients. And this can have implication to the prevalence of Schistosomiasis in the area. So aside from the water, we should also look into the soil samples because these snails could estivate and hide in the soil for quite some time. Okay. Now for the last part, let us look at the genetic studies. Okay. Now the first one, we looked at the overall genetic diversity of the Oncomelania hupensis quadrasi found in the Philippines vis-a-vis other subspecies. So this was a a part of the MS thesis of James Christopher Chua. Okay. Now, what we found out based on the 16S and which has been corroborated based on other markers, OH quadrasi is a distinct group. And OH quadrasi is a distinct group with at least four different haplotypes. And if you're going to look at the distribution in the three of the different subspecies of Oncomelania hupensis, it appears that there's a need to reevaluate the taxonomic designation of these subspecies, precisely because even the Chinese isolates are found to be polyphyletic. They're found all over the place. Okay, so for example, OH hupensis and OH nosophora, which is found in Japan, they seem to cluster here. Or actually, they don't have a distinct topology. And then this one is a polytomy, so there's no clear structure. And identifying subspecies based on sheer geography may not be the best way to identify or to designate the subspecies of Oncomelania hupensis. In fact, based on our results, we found out that the genetic distance among the different subspecies seems to be so great that they can in fact be designated as different species and not just subspecies of a species. Okay. And then furthermore, when we continued the DNA barcoding of the different strains of Oncomelania hupensis quadrasi found in the Philippines, one of our students, Elaine Claire Macaspac, together with Rafi Cornelius, discovered a potential new species of Oncomelania here in the Philippines. So when we compared the genetic distance, so we compared the intraspecies and the interspecies variation. So how if you compare the differences with of individuals of the same species, in this case Oncomelania hupensis, and then those of different species, there is a distinct genetic barcoding gap. So the distance within species significantly smaller than between species. However, if you're going to look at the differences with between different subspecies, the genetic distance is still very great, to the point that you can assume that the differences are the same as those found between species. So this is again another point for taxonomic evaluation. Should we still consider the different subspecies of Oncomelania hupensis as subspecies, or should we now elevate them to different species? And then furthermore, we found an unknown individual, an unknown population of Oncomelania hupensis, supposedly Oncomelania hupensis quadrasi, which has a very big genetic divergence compared to the other Oncomelania hupensis quadrasi here. So the genetic distance is in fact so divergent to the point that they could be regarded as different species. This is despite the fact that morphologically, or based on the shell, they are very, very similar. Now, this has some implications, particularly if you're going to put it within the context of susceptibilities. If you have different subspecies of Oncomelania hupensis, or in fact, different species of Oncomelania, instead of just Oncomelania hupensis subspecies, we could elevate them to different species, then we should also be concerned with the differences in susceptibility, so different strains of Schistosoma japonicum, as discovered by Rashford in 1977 and Cross et al. In 1984. So when they compared the susceptibility of the different strains of Philippine Oncomelania hupensis quadrasi against different strains of Schistosoma japonicum, as well as a different subspecies of Oncomelania hupensis against a different strains of Schistosoma japonicum, are they comparing taxonomically distinct species? Or maybe the variation in the results could be attributed to variation also within the same species or the taxonomic operational unit that they are using. So again, there is a need to review the susceptibility of the different strains of different subspecies of Oncomelania versus the different strains of Schistosoma japonicum, but this time within the context of the genetic variation, based on their supposed correct taxonomy. Okay, so in summary, the distribution of snails and infection rate are patchy here in the Philippines, which is to be expected. as seen in the differences, significant differences in the habitats that they found in the Philippines. Environmental variables also affect the distribution of the snails. And also, eDNA is a viable approach for biosurveillance, and genetic diversity of snails could reveal more than one subspecies or even species here in the Philippines. So moving forward, what could be done based on what we have discovered? There's a need to optimize protocols for biosurveillance, a means to disrupt the life cycle of the parasite. So knowing where they are is very important in the is critical. Knowing where the snails are is critical, especially in the control and elimination of the parasite, particularly schistosomiasis. And excuse me. And eDNA is a viable and safe option for biosurveillance. Then next, there's a need to examine more closely the effects of climate change on the dispersal of the snail, especially since the effects of climate change is becoming more and more pervasive and prevalent. And lastly, we need to re-examine susceptibility of different subspecies of Oncomelania hopensis against different strains of Schistosoma japonicum based on their genetic distinction. So with that, I would like to thank the following. This is a collective effort of everyone from our universities and from our university and also from our collaborators. And we hope that we can continue our collaboration in the foreseeable future. And so thank you very much. And I will be more than happy to entertain your questions.
Wow, this is really game-changing work by yourself and your team, Dr. Ian. It's really interesting as well to see that you mentioned climate change in relation to schistosomiasis, in relation to disease, has been mentioned several times, both in Dr. Lydia's work and in your work. And we really look forward to hearing a bit more from you during our discussion on what this could mean for species, subspecies in other areas of the world and what we can learn and transpose from this. So thank you very, very much, Dr. Ian. But for now, I'll hand over to you, Dr. Mario. We don't have a recording for you. You're presenting live. And
Yes,
so on that note, I will hand over. And thank you for joining us.
All right. Thank you very much to the Global Schisto Alliance, as well as the International Society for Neglected Tropical Diseases for this opportunity. I'm going to share with you our experience and hopefully some progress on vaccine development for Schisto japonicum. Again, I'm from the Research Institute for Tropical Medicine of the Department of Health. And this is basically a collaboration with the Center for International Health Research at Brown University, with my mentor, Jake Curtis, as well as with Dr. Hannah Wu. So I think the Philippines is one of the first countries to demonstrate that mass drug treatment and mass drug administration alone is difficult to sustain schistosomiasis control, because data as far back as 1981 has shown that, if we tend to stop or at least delay our mass treatment programs, the baseline prevalences would just return to their usual numbers. As you can see here, from 1981 to 44% prevalence in one stool catocats. When we returned in 2002 in the same village, we found an 82% prevalence of infection. So I guess the difference is just because a three-stool catocats is more sensitive, but nonetheless, it's sufficient to say that prevalences can just return back to their usual or their previous numbers if we stop MDA or if we discontinue our control programs. And when we returned back in 2016, the prevalence was 53% in the same village. And back in last year, before the pandemic started, we returned and the prevalence was still 47%. So there's still a persistent hotspot infections in the Philippines if we do not have continuous access to mass drug administration. And the other challenge in the Philippines is the very high levels of reinfection that we see in our communities. Seen here in the 2002 cohort we see 82% reinfection after 12 months and up to 90% reinfection after 18 months. And so there's very rapid reinfection because, of course, of water contact. And as what Professor Leonardo mentioned earlier, water contact is part of their daily lives in rice farming and washing their laundry and doing their dishes. And thankfully, probably because of mass drug administration, in a span of about 15 years, the reinfection rate after 12 months has been reduced to about 30%. And last year, it was about 54% reinfected after 18 months or a year and a half. So suffice it to say, from the last two data here, if we do not continue mass drug administration for 2 or 3 years, we just expect that everybody that was infected before was just going to be reinfected. So with that, it's very difficult to sustain the progress in reducing schisto prevalence if we are just dependent on mass drug administration alone. Therefore, there is a need for alternative control strategies to sustainably control schistosomiasis, particularly Schistosoma japonicum here in the Philippines. And one of those most promising avenues for alternative control strategies is vaccine development. And in our work for the past 20 or so years, we've tried to understand the immune response of humans into Schistosoma japonicum, and we look at treatment reinfection studies where we look at their antibody levels at baseline, and then we follow them up for a year and a half, and see if these antibodies predict resistance or susceptibility to infection. And what we published a long time ago was that a vaccine candidate called SJ97, which is a paramycin or a muscle protein, we found that the individuals who had IgE antibodies to paramycin had the lowest levels of Schistosoma japonicum infection intensity 12 months after follow-up. So IgE antibodies specific to SJ97 predicted resistance to reinfection. Similarly, IgE responses to another vaccine candidate, SJ68, as well as SJ41, were also associated with reduced reinfection intensities, showing that they may be predictive and they may be good vaccine candidates. However, the caveat is, as in all helminth infections, IgG4 responses make us all susceptible to reinfection. So we just published a paper this year on a new set of vaccine candidates using differential screening, either by phage display or cDNA cDNA screening. And we have a new list of vaccine candidates that we can play on, that we can pursue in various preclinical studies. And among the different vaccine candidates, the most advanced of all is SJ97 paramycin, because we have developed a pilot scale production process using E. coli to produce recombinant S. japonicum paramycin. And as you can see here from the E. coli lysate, we had several purification steps until we get about 95% purity, which is sufficient for vaccine development. And in initial studies in large animals, we have demonstrated that this vaccine, recombinant SJ97, is immunogenic and can induce antibody responses, as seen here in the ELISA of SJ97 plates. And more importantly, our colleagues from China, from the Shanghai Veterinary Research Institute, have used this vaccine candidate, this recombinant protein, to evaluate vaccine efficacy in the water buffalo, because in China, the water buffalo is a major transmission agent, and it spreads. It's a major factor for the transmission of Schistosoma japonicum, especially in the rice fields in China. And what they have found out is that vaccination with recombinant SJ97 led to about 50 to 60% reduction in worm burdens after they challenged these water buffaloes with Schistosoma japonicum, of course, the Chinese strain. To note, though, this vaccine candidate was identified using the Philippine strain of Schistosoma japonicum paramycin. And it's nice to know that even though the Philippine strain SJ97 works well with the Chinese strain S. japonicum. So in the Philippines, of course, we wanted to replicate this work as well. And we wanted to know if this SJ97 vaccine is also protective of schistosomiasis infection in large animal models in the Philippines. And as you may have heard, Schistosoma japonicum is unique because it is a zoonotic infection, and it infects as many as 46 mammalian hosts, not only the water buffalo, but every domestic animal you can think of: dog, cats, pigs, field rats, rice field rats, and cows. So all of these mammals can be infected with Schistosoma japonicum. But we are very much interested with the water buffalo because, one, the studies in China have demonstrated, again, that the water buffalo is the major transmission agent. So we also presume that this is also the case for the Philippines because we have also so many water buffaloes here in the country. And at the same time, you know, the interaction between humans and water buffaloes in the rice fields is very intertwined, that it would not be surprising that the water buffaloes would also be the major transmission agent in the Philippines. But there is very limited data in the Philippines when it comes to water buffalo prevalence and transmission. So what we initially did was to try to assess if water buffaloes in the Philippines are susceptible to Schistosoma japonicum infection. And we have published this paper showing there is indeed high prevalence of schisto in water buffalo. Because what we did here was that we got 34 water buffaloes coming from a non-endemic area, so they were negative for schisto, and then we transported them to an endemic village in Leyte so that they will be exposed to schisto, so that if ever the animal or the water buffalo is susceptible to schisto, we will be able to determine through various laboratory tests if they are indeed infected with schisto. And this is also during the time of Typhoon Haiyan. So instead of the original 6 months plan, it was extended to over 10 months of exposure. But based on and this is what we did after 10 months, we collected all the 34 water buffaloes again, and we did necropsy so that we can perfuse the Schistosoma japonicum worms outside of the mesenteric veins where they are lodged. As you can see here, we are trying to collect the worms. And these are the Schistosoma japonicum worms that we collect from the sieve from these water buffaloes. And if we look at the data, out of the 34 water buffaloes, only one was negative for Schistosoma japonicum, and 97% of them, of the animals, had Schistosoma japonicum worms with a mean of 100 adult worms in each animal, showing that water buffaloes are indeed a major transmission reservoir for Schistosoma japonicum in the Philippines. It's interesting that if you compare the perfusion with a coprologic test, the formalin ethyl acetate sedimentation technique, as well as real-time PCR, these two techniques somewhat underestimate the prevalence of infection. They can only approximate, the prevalence of infection if you do multiple tests of FASD or multiple tests of qPCR sampled over a span of 3 to 6 months. So you need to have multiple samples in order to do that. So from this data, we know that water buffaloes are a good ideal model for Schistosoma japonicum infection. So initially what we did was to do a vaccine evaluation in carabaos that are already naturally exposed to Schistosoma japonicum. So what we did was that it was a placebo-controlled vaccine trial with 20 carabaos vaccinated and another 20 carabaos that were placebo. And they were vaccinated in an area that was negative for schistosomiasis, so they were not infected at this point. And when we were vaccinating them, there was no anaphylaxis or severe hypersensitive reactions. And then afterwards, we distributed these water buffaloes, if you notice them, in pairs, because one of this animal is a vaccine a recipient and one of them is a placebo, so that they should have like a similar exposures. And then we allow these animals to stay in an endemic village for 6 months because we expect them to have some Schistosoma japonicum infection after 6 months. But of note, at baseline, even before they were vaccinated, we already noticed that most of the animals had high antibody titers to schistosoma worm antigen to swab. So this might be a complication in our future analysis. Okay, so during vaccination, we monitored their blood counts, their CBCs, and we found no differences between vaccinated and placebo animals in their white blood cells, RBCs, platelets, as well as hemoglobin, suggesting that the vaccine is safe, at least in the short term, 1 week after the dose, and in the medium term, 4 weeks after the dose. So again, suggesting that the vaccine was safe in these water buffaloes. And the vaccine was very immunogenic. If you can see here, the mean titers of the vaccinated animals versus the placebo animals. The placebo animals did not have any antibodies to SG97, but those that were vaccinated had very, very high antibody levels, particularly IgG1 here in red, and also some IgG2 titers. And although they were very immunogenic, unfortunately, we did not see any protection in these animals because there is no statistical significance between the worm burdens of these animals comparing those that were vaccinated and were placebo. And there were challenges because of differential exposure. Even though they were matched pairs, we were not really sure if they have the same exposure to the same number of parasites. So the next set of experiments we did was to actually establish a water buffalo model for experimental Schistosoma japonicum infection so that we know exactly how many Cercaria or parasites we're going to challenge these animals during vaccination. Okay? And so in order to establish this water buffalo model, we needed to have a water buffalo facility. And we established one in Padre Garcia in Batangas because Padre Garcia is a town which is known for an animal auction town where animals, not only water buffaloes, but also cattle, as well as goats and other animals, are being brought to Padre Garcia for auction. And so we can select animals coming from non-endemic areas here in Padre Garcia, and we can just house them in our water buffalo facility. Okay. But the other challenge really that we found with the SJ Japonicum challenge infection experiments was the difficulty in obtaining as well as in finding infected Oncomelania hupensis quadrasi snails. As what Dr. Ian has just described earlier, these are very, very small snails here. This is the smallest coin that we have in the Philippines, a 25 centavo coin. And the snails are a bit just 1/10 of that, and they're very, very small. And so unlike the other intermediate hosts that can easily shed in the presence of heat and light, Schistosoma japonicum does not shed outside of Oncomelania quadrasi. So you have to crush these snails. You have to sacrifice these snails in order to get Cercaria. And they're also very sensitive. They get inactivated. The Cercaria get inactivated if you expose them to more than 30 degrees Celsius. So we have to do these experiments at night so that we have cooler temperatures because we are, of course, in the tropics and we are in the Philippines. And the field snails that we collect are only 0.5% positive. And if we reinfect them in the laboratory, we can increase them only up to 3.7%. And we need more than 500 Cercaria per animal. So we need tons and tons of these snails. And so, and we only get to see about 30 Cercaria per infected snail by crushing. So the initial challenge really that we had was that we had a lack of Oncomelania snails if we are dependent on field collection of these snails for our vaccine challenge experiments. If these were just mice experiments, we only need 50 Cercaria, then this would not be a problem. But we are using the water buffalo, and you would need hundreds of Cercaria, so this would be a big challenge. But nonetheless, we tried to pursue with our vaccine work. If you notice our small sample sizes because of the limited snails, but we tried first SG97 in various adjuvants. Of course, we started with ISA206, the same the one that we use in China. But we also tried alum or alhydrogel, commercially known as alhydrogel, as well as MPLA or monophosphoryllipid A, which is an TL4 agonist to excite our immune systems in coordination with alum. And what we found out is that their IgG levels are significantly higher compared to placebo, especially those that were from immunized with ISA206. And most strikingly for IgG1, the fold increase in antibody titers was also much more evident in the ISA206 group. But when you look at the perfusion, this is a perfusion after infection with, this is 500 Cercaria for each animal, we found out that it seems like that MPLA is a very good adjuvant because we did not see any worms in those animals, in those water buffaloes that were immunized with MPLA and alhydrogel, compared to those that were immunized with ISA206, SG97, as well as SG97 and hydrogel. But one thing that I want you to focus on is the number of worms that we are able to collect after perfusion. See, from an initial dose of 500, we only get to collect from say 6 to 10 worms. So there's a very, very low yield of development from Cercaria going into adult worms. So since we found that MPLA was a good adjuvant for SG97, we repeated this experiment exclusively with just MPLA and comparing it with placebo, this with just 10 animals, five carabaos in each group. And when we did perfusion of these animals, we found out that there was no statistical difference between the two groups. So we did not see any protection. In fact, SG97 vaccinated animals had quite some degree of higher worm burden, but this was not statistically significant. So in effect, the observation we found here was not consistent with the repeat experiment that we see. Okay, so otherwise, we also continued with another vaccine candidate known as SJ68. And so we looked at SJ68 formulated with, again, MPLA and alhydrogel alone, just by itself, or in combination with the SJ97. And when we challenged these animals with 1,000 Cercaria of S. japonicum, what we found out is, again, there was no statistically significant differences in their worm burdens, suggesting that there was no protection. Although, again, with 1,000 Cercaria, we now increased the dose to 1,000 Cercaria because initially the worm numbers were very low. So even though at 1,000, we only see about 20 to about 40 adult worms after perfusion. So still, the development or the yield from Cercaria to adult worms was relatively low. Okay, so that was a consistent challenge among the different animals that we see in the different groups, that the worm patency or the development from Cercaria to adult worms was very, very low, less than on average is about 2.5%. And so we tried to understand what was the reason why there was very, very low development into adult worms. And so we designed an experiment where we tried different types of animals, no vaccination at this point, just trying to look at what factors would contribute to increasing the number of adult worms in water buffaloes after challenge infection. So we looked at, this is our control using 1,000 animals. Then we had a high dose. We actually used 2,000 Cercaria as a challenge dose. And then we also tried, looking at the natural infection. They're not being infected one at a time. So we used trickle infection. What we used was actually 4 times 500 Cercaria challenge. So every week we would challenge the animals with 500 Cercaria. And also we tried doing an earlier perfusion. Instead of 8 weeks, we would do 6 weeks infection. And we also tried not treating them prior to vaccination or prior to challenge infection to see if praziquantel or albendazole has an impact in the development of the worms. And also trying to look at more or younger animals, juvenile water buffaloes, if they were also susceptible. And finally, we also tried to see cattle, if cattle are good animal models, although cattle is not as ecologically relevant for Schistosoma japonicum compared to water buffaloes. And here is our results. You can just look at here the average recovery. On average, we see somewhat an increase in worm yields if we do treatment C, which is trickle infection. So this is 500 Cercaria divided into several infections. So that somehow increases the yield of adult worms that we collect after perfusion. And surprisingly, the group that had juvenile water buffaloes, 1 to 1.5 years old, had also higher worm yields. So comparing that to the normal ones, where the initial dose of 1,000 will give you about 50 or so or 100 or so worms. Here, the 1,000 worms will give you up to 140, 150 worms afterwards. And cattle was also a very, very good model for Schistosoma japonicum, but we decided not to continue with cattle because, again, it's not somehow relevant for the transmission of Schistosoma japonicum, not as important as the water buffalo. So the current experiments that we have are using, consolidating all the results that we had from previous studies. So we're now using young carabaos, 1 to 1.5 years old at the start. And we're using now trickle infections, using 4 by 500 Cercaria weekly. So we did infection over a month every week. And we are now using snails from the Biomedical Research Institute in the US because, again, of the sustainability of getting infected snails from the field. So we have been collaborating with BRI for the last 2 or 3 years. They've helped us establish our snail colony at RITM. And so they were more than gracious to give us their Schistosoma japonicum snails, as well as the infected snails that we can use for our water buffalo studies. And this is still ongoing with 3 groups of 8 animals each, vaccinated with just SJ97 or SJ97 and SJ68, or placebo using ISA 206 as our adjuvant. And as expected, we see very good, robust immunogenicity after 3 doses here in purple for IgG1 as well as for IgG2, as well as also for SJ968, very good IgG1 responses. And so we look forward to the perfusion that will happen probably the next month or so in December. We'll see, hopefully, if we get any protection from these animals. So in summary, there is still a pressing need for new interventions to sustainably control schisto in the Philippines, and one of which is vaccine development and a promising alternative control strategy. And we have identified many, many vaccine candidates that we can utilize in our animal models. The water buffalo is a very appropriate animal model for Schisto japonicum. And we've demonstrated that SJ97 and SJ68 vaccine candidates are safe, as well as inducing robust antibody responses, and that juvenile water buffaloes with trickle schistosome infections offer promising infection yields that are hopefully sufficient for vaccine trials. So with that, I would like to thank all our collaborators as well as our team from Brown University, Dr. Jake Curtis and Dr. Highway Wu, as well as from SBRI, who helped us with perfusion, necropsy, as well as infection of water buffaloes. Of course, from the BRI for the snails, and the staff, the study participants in our cohort in Leyte, funding from the NIH as well as WHO in my early work, as well as our staff in RITM, both in Leyte as well as in Metro Manila. With that, thank you very much.
Thank you very much, Mario. Sorry, my video still hasn't come back on, so I'm quite uncertain as to if you can hear me or not. I'm judging from the smiles.
Yes, we can hear you.
Okay, so that's good news. Well, to our three presenters, a huge thank you. I think our attendees will absolutely join us in saying we've been completely blown away by the depth and the breadth of the work across the Philippines. We've heard about so many things in the last hour and a bit, whether it be possibly new species all the way to a tremendous potential for the vaccine you've told us about. We've heard about climate change, the impact of COVID, and how to overcome this. So it's been an amazing session, and beyond that, just a huge body of collaborative work coming from the Philippines. And it's been really interesting being able to focus on the country experience in one particular place, because we can really see how and when you've collaborated and so forth. And I think that the first thing perhaps that we'd like to ask you, I suppose as three researchers, of course, you know each other very well, you've co-authored papers and collaborated a lot, but all the different sectors that you're coming from, have you found that there's been sufficient collaboration and dialogue between all your different organizations or groups, or even just sort of broader fields of study? Perhaps, Lydia, I could ask you first.
Yeah. Okay. I think there have been a lot of researches in the Philippines covering various disciplines, especially with when we had this Eco Health project, where we brought in a lot of people from different disciplines. So I'm sure that if we really just continue with this kind of approach, bringing in people from different disciplines, not just on biomedical, but also from social science, behavioral science, and even economics and culture and everything, and environmental science for that matter, aside from biology, not to mention biology, of course. I think we will even be doing a lot. And in fact, the people that you see now, myself and Ian and Mario, we have been working on this, and we're really trying hard to involve other people as well. So whereas before, I remember Mario and myself, RITM and CPH, I think College of Public Health and University of the Philippines, Manila, and RITM, I think among others, these are the two institutions that are really doing intensive work on schistosomiasis. And then eventually we have involved the University of the Philippines in Diliman, and in fact even Los Baños, other campus of UP is also being involved in this, and even other campuses down in the south, in Mindanao, and even in the Visayas. So I think we're I think the interest is what you call this, snowballing or spreading actually, and intensifying for that matter.
Fantastic. Ian, would you like to add something to Lydia's words?
Well, actually, I'm a perfect example of someone I never thought I'd be working on schistosomiasis. And I'm a geneticist by training, and I really look at evolutionary biology and malacology. And then I'm not going to say I was yanked into this project, but I got involved with so many people. And then eventually I got linked with Dr. Lydia Leonardo through the Malacological Society of the Philippines, and my interest was primarily on snails. So that's how I got involved. So just to emphasize the fact that the schistosomiasis study in the Philippines is multifaceted. multi-disciplinary. And then we hope that we will also involve other areas, for example, the economic aspect, tying the economic aspect with regards to snail control. How can we make planting rice viable while at the same time, or is there any other way we could do to modify the environment of the snails without affecting the economic aspect of our farmers? So it's a multifaceted thing, and we hope that we will bring in more people with different disciplines into the table. And I'm also happy that Mario is also making great strides uh, in the in the vaccine department. So I don't know if he's moving to the VIP soon, but we'll see.
Definitely.
I'm sorry, VIP is Vaccine and Virology Institute of the Philippines. It's a new institution. Yeah,
Maybe both kinds of VIP.
we have
Very
both kinds of
important
VIP.
person and VIP. VIP at the
Yeah.
VIP. Mario, any thoughts from yourself?
Well, I'm not a virologist. I'm definitely not part of the Virology Institute, although I'm being dragged with COVID-19. But nonetheless, the groundwork has been laid. There's a great network for schistosomiasis research in the Philippines. Dr. Lydia Leonardo, in collaboration with our late Dr. Olveda, as well as my mentor, Dr. Luz Acosta, they were all great schistosomologists, and so they paved the way for researchers to be engaged with schisto. And interestingly, as we keep on banging the drum to tell everybody that schisto is still a relevant public health concern in the country, a lot more people are getting interested in schisto. So within the country itself, there has been tremendous interest in schisto, as what I've observed. But on top of that, we Filipinos love to collaborate, and we love working together, and we love going, to meetings and conferences and partner with each other. So that's a great avenue for us to network and to partner not only with local but also with international collaborators.
Exactly.
And I think moving forward, schisto will not be a neglected research priority in the Philippines as long as we get support and we get funding. There are a lot of people that will be very interested to working with schisto.
Absolutely. I think as you've presented it today, it's made a really strong case, and the buy-in from those funders and those policymakers. So that's really positive. I suppose the one thing that kind of brings all of these things we've discussed together is the concept of One Health. And I just wanted to ask how you found working perhaps with ministries of agriculture or those on the animal health side of things, and also could a parallel be drawn with, for example, sleeping sickness, where there was actually a very strong incentive for farmers themselves to fund the protection of their cattle against the tsetse fly? Could we see a similar dynamic with the One Health and buffalo approach? I read more than 50% of buffaloes can show signs of infection, so that's quite a high burden. And this also ties in with a question here. We've got lots of questions now coming fast and thick on our chat. A question from Anouk Gufras from the GSA, just asking, are there opportunities for a One Health approach with water buffalo vaccinations in the future?
Yeah, if I may start, for the water buffalo scenario in the Philippines, it's very unusual, unlike in China, that the water buffaloes here do not actually get sick from schistosomiasis.
Exactly.
They're very healthy. They're very plump and healthy, even though they have hundreds of worms inside of them. And in fact, I'm pretty sure that the water buffaloes that are being slaughtered for meat are infected with schisto because they do not see any signs of morbidity from parasitism. They do have if these animals are infected, say with fasciola or other rumen flus, they can get serious infections, or even with Sura. Sura is a major leading cause of death for water buffaloes, but not schistosomiasis, unfortunately. So it's quite a hard call to bring in stakeholders from the animal industry. However, our common point is our farmers, because our farmers are very susceptible to schistosoma infection if their water buffaloes are infected. So it has to be a holistic approach where even though your water buffaloes may not be sick of schisto, your farmers may be getting sick of schisto and may get hepatic fibrosis and other complications if you do not do an integrated One Health approach. Ma'am Lydia, do you want to
Yeah.
add something?
Yeah. In the past, it was very difficult to get on board the Department of Agriculture and Bureau of Animal Industry, but because of the persistence of the Department of Health, that and just like what you said, that we're talking about the health of farmers here who are going to use the carabaos, and the carabaos, even if they don't show any serious morbidity, but since they contribute significantly to transmission, then something has to be done. And I think what really convinced the Department of Agriculture and Bureau of Animal Industry to get on board. And then, just like what I said in my presentation, that there's going to be a national policy on involving the Department of Agriculture and Bureau of Animal Industry. Bureau of Animal Industry in like really developing a program that would address the involvement of carabaos in transmission. And I think we're getting some headway into this.
Fantastic. Ian, would you like to add anything?
Well, I could speak from personal experience, and when I say personal experience, this is an experience I shared with Mom Lydia. We have, when we go into research, when we go to field work to different sites, especially to new endemic areas, it's always a problem coordinating with different government agencies, the Department of Agriculture on the one hand, and then the Department of Health. And then sometimes we even have to go to the Department of Environment and Natural Resources for environmental collection. So it has been difficult before, but now I think Mami is right. There is some headway, especially if we're going to have a national policy that will coordinate all these agencies. They are now familiar with us because we are very, very persistent with our buying them their offices. We need your permits. And we have to explain to them every so often why we have to do this in an integrative approach. Because if you're from government, it is your tendency to, that's not the jurisdiction, go to the other office. But the thing is that we have some headway. We have been able to show them the significance of working together. And in most coordination meetings, we always involve these government agencies. And it does help when these agencies talk with one another to facilitate our sampling, our processing of samples, our processing of permits, and they also understand what we are now doing. So hopefully we're making some dent here when we talk about banging the doors of the government. Just have to be persistent.
Exactly.
We hear that advice. Fine. Notes taken. And it's true. I mean, it helps a lot. These are just practical considerations, I suppose, in parallel to the science, but ultimately what's needed to move the programs ahead. So thank you for those insights. We had totally underestimated the session, allocating 90 minutes for it. And we're kind of reached the 90-minute mark. However, there's more and more questions, of course, elicited by your incredible work. And so I was wondering whether it would be okay to stay on for a little bit longer.
Yeah, sure. No problem.
Super. Thank you so much. So, well, I'm not sure where to start with. There's questions here ranging from climate change and, of course, around the vaccine. So perhaps just going back to the MDA, for example, a question here from David Rawlinson. The MDA program is really critical for schisto control. Is the program aimed at all age groups?
The MDA is supposed to be aimed at all age groups, but since it's really very difficult to involve all the population, it's supposed to be covering 5 to 65 ages, 5 to 65. But I think the work is really to make things easier and to make sure that everything is going to be consistent. There's a specific group that is being involved, and which are the school children, because they are easier to manage, and MDA is easier to implement among school children because at a particular time, like they are there, and the teachers can take care of supervising MDA, of course, with the assistance of doctors and the nurses as well. And the other thing is, school children are also very much exposed to schistosomiasis, but there are also I think programs for the entire community. So there's a program of MDA for school children and another program also for the community. So there's for school children and the specific schedule for the community in order to be able to cover the age group, which is from 5 to 65.
Fantastic, thank you. There's quite a few questions here from Bonnie Webster from the Natural History Museum in London, which holds quite a large schistosome collection. So perhaps the first one would be, interestingly, actually, Dr. Mario, Bonnie Webster saying lovely work. And are the S. japonicum vaccine candidates relevant to the African schistosoma species?
As I mentioned earlier, our clones for the recombinant production of the vaccine were based on the Philippine strain of Schistosoma japonicum, but surprisingly, they were effective for the Chinese strain. So we are also hoping that we haven't done any formal evaluation yet, but we're more than happy to collaborate with our African partners to assess if these vaccine candidates can also be protective of Schistosoma mansoni or Schistosoma hematobium. Although it might be a challenge as to what animal models we will have to use, because unlike Schistosoma japonicum, which is a plethora of animal models, I think we have a much limited zoonotic nature for the African schistosomes.
Fantastic. And another question on the SJ97 vaccination. Does this have any impact on egg production or fecundity?
Well, we initially are doing evaluations on just worm counts, but we are going to evaluate as well liver egg depositions, and so that data will be forthcoming.
Thank you. Well, we're definitely going to be watching this space. And question again from Bonnie Webster at the NHM for Dr. Ian. Very interesting and lots of complexity with the snail species. Is there any evidence for different compatibilities between different snail strains and S. japonicum from different hosts?
Yes, actually, there has been several studies in the 1970s and 1980s that compared the different susceptibility or rate of infection of the different strains of Schistosoma japonicum versus the different subspecies of Oncomelania hupensis. However, that was then I think the last one was 1984. So we need to revisit this within the context of their genetic strain. As I've mentioned, there's a big question mark on the validity of the taxonomic designation. So we're not even sure if the studies that conducted if the studies that conducted these susceptibility tests were using the appropriate taxonomic designation of the snails, and well, I'm not sure about the Schistosoma japonicum, but definitely the strains of the subspecies of Oncomelania, so hupensis. So we need to revisit this, and I personally have not conducted this. It's been more than 3 decades ago, so I think it's something that we need to do, and yeah.
Wonderful. So please come back when
We've done that.
Yeah. Another question here from David Rollinson. Any thoughts about snail control and linking with the new surveillance methods?
Well, I could speak in behalf. I could speak from the perspective of eDNA. eDNA is a very easy method to use. But one thing that we should bear in mind is that if you use eDNA, it only tells you that the eDNA is there. It does not necessarily mean that the actual specimen is there. It could be that the eDNA came from elsewhere, or it could be that the species or the snail or the parasite has long been dead, and what you're detecting is the eDNA, and we don't know how long that specimen has been dead. We still have to evaluate the validity of eDNA with regards to its life, shelf life or lifespan in the environment. Will it last for a few days or a few weeks, etc.? So just what I would like to emphasize, that the negative result in eDNA does not mean that the specimen is not there or the species is not there. So that is a caveat that you have to be careful with. Be that as it may, the eDNA method can be used to complement the malacological survey, at least from the perspective of the malacological survey. And if we know where the snails are, we can identify them as potential risk areas, and perhaps we could do some modification to the environment to prevent to limit the dispersal of the snails, or to prevent or to kill them. It's quite difficult to kill them or eradicate the snails completely, especially since they're very small. But we can limit their dispersal. We can modify the environment, like cementing the what do you call
Irrigation
the
canal.
embankments, irrigation canals, or we could convert rice fields to orchards, like what they did in Japan. But the economic impact of that still has to be evaluated. Filipinos are voracious rice eaters. I don't know what will happen if you're going to economically dislocate our farmers. If we're going to convert the rice fields into orchards. So what I'm saying is involves many disciplines in order to address this question. But definitely we can use the eDNA method as a complement to the malacological survey in order to control, as a biosurveillance and control of the dispersal of the snails.
Fantastic. And building on that, another question, are there natural predators for the snails? And if so, does habitat change or climate change affect predator densities, leading to increased transmission?
That is a very good question. We have always been wondering about that ourselves. The snail is very, very small, as you can see.
Exactly.
Sometimes you go into the field work, if you're not an expert, you could easily mistake them for a grain of dirt or soil or mud. So I'm not sure if they are known predators, at least for huge vertebrates. I'm not even sure if birds would notice them.
Exactly.
So yes, at the moment we don't know if there are known predators and whether they will be affected by climate change. But definitely their dispersal can be affected by climate change.
That's so interesting.
Yeah.
Thank you.
Ma'am Lydia would like to.
Yeah.
Of course.
Exactly. There was a time that I mean, like, being very practical about it. There were some people, or I'm not sure who these people are, who were suggesting that let's use ducks. Let's put ducks in the rice fields, or ducks in swamps, and let these ducks eat this oncomelania. But we were saying that these are so very, very small snails, and the ducks might not even be able to see them, let alone feed on them. So that strategy was eventually abandoned. And in terms of transport of snails, I think the ease with which snails can be transported is already known, and we have seen that particularly in Gonzaga at the present time, such that initially when Schistosomiasis was discovered in that area in the south, there were only three villages that were endemic, and then just lately, maybe like just 5 years ago, there were two new villages that were added into the list, and like, people are getting scared now, not just from that municipality but also in neighboring municipalities, because the connection is very intense in terms of river networks and irrigation networks, and really fear that those in high elevated barangays might transport or spread the snails from their areas in the low lying barangays, which is actually what has happened in the past.
That's really fascinating, very different to other areas. So thank you for telling us all about this. Bonnie Webster, who asked many of the questions, is saying thank you for the great presentations and discussion. And we'd like to thank you for your amazing questions as well. And thank you, David Rollinson, too, and all who participated. I think we are coming towards the end of the webinar now. And so perhaps if I could just hand over to you one final time, Dr. Lydia, Dr. Ian, Dr. Mario, what would be your sort of message to our international audience, whether it's the Schistosomiasis kind of network around the world, or just more broadly the global health and neglected tropical disease field? What would be your thoughts for future partnerships or future steps for schisto control?
Yeah, I think what I can say is we're all in this together. We are fighting the same disease, which is schistosomiasis, whether it is in Africa or South America or the Philippines. The disease is equally debilitating. The disease is equally causing a lot of morbidity in people that they affect. So I think the best way is like what we're doing right now. We can exchange notes, we can exchange experiences. Maybe we can benefit also from the experience of our African partners, and then maybe put this all together, and then we'll come up with like a battle plan against global schistosomiasis or something like that. So I think that's my takeaway message for everyone.
Dr. Ian, what about you?
Oh, yes. As we all know, the schistosoma has a very complex life cycle, and therefore this gives us a glimpse of hope in order to address this. There's no one single solution for this. It's multifaceted. We can target the parasite at its various life stages. There are various ways by which we can control the disease. We can look at the snail intermediate host. We can look at the reservoir host. We can look at mass drug administration, simple wash method, and then vaccination. So there are several ways by which we can address this. And for each area, we can have a whole avenue of research. So this is going to be a global or pan-tropical or even global effort. So like what Ma'am Lydia said, we're all in this together. So we should help each other.
Fantastic.
Thank you.
Thank you. Dr. Mario.
Right. So I hope we have given you an insight as to the burden of disease, as well as the research studies on the Asian schistosome. And so we hope that Asian schistosomiasis will be further be engaged in different international venues because for now, we have very limited exposures. So we do hope that Schistosoma japonicum will get more international mileage, and we do hope for further collaborations with everyone. And with the uniqueness of Schistosoma japonicum, I don't think any more disease is more fitting for an integrated One Health approach, because we need to have the animal sector, the vector control sector, the human MDA sector, and even environmental sanitation and all that integrated as one, so that we can address the concerns of schistosomiasis. I have been asked over and over again that schisto has been a persistent problem for more than 50 years, ever since our institute has been established. And if we do not do anything in our lifetimes or in the future, it will persist to be as one. So we need to have a better approach towards its sustainable control and elimination.
These are perfect concluding words. Persistent, recurring schistosomiasis hotspots is something that is seen around the world. So I think your three presentations have been amazing within a very short amount of time, not just stating that we need a One Health approach or we need all these different fields working together, but really demonstrating that really, really clearly with your experience from the Philippines. Thank you so much for sharing this. It does sit into the broader and kind of wider GSA and ISNTD collaboration on this series of webinars around schistosomiasis. So anyone who's tuning in for the first time today, please do go. We've got all the recording on a YouTube channel, on a playlist, and with examples, as you just said, across the African continent and kind of around the world, and lots of different approaches. So thank you really for bringing all that together. What's really striking about working with anyone from the Philippines is not only do you have a beautiful country, but just the energy coming out of the researchers and the collaboration and the teams. It's really wonderful. It's really infectious. And this is always the case when we have the privilege, the pleasure of getting to meet with you. So thank you very, very much. Thank you also to Anouk Gubras, who was on the chat posting the right resource at the right time. That was really very helpful, fantastic. And thank you to all of you who tuned in today. I've thoroughly enjoyed the session. I'm sure you have too. Lots of thank yous on the chat. And Lydia, Ian, Marco, we really look forward to keeping in touch and having you back on this platform very soon with updates, and we'll be turning to you for inspiration. So thank you so much. And hopefully see you very soon. Salamat.
Yeah.
Salamat.
Thank you.
Salamat
Thank you very
din.
much.
Thank you very
Thank
much.
you very much, Mara.
It's
Take
been our
care.
pleasure. Thank you.